So in the previous class, where did we left off? I believe we have done the... Okay. So we were talking about the plasma membrane, isn't it, students?
Yes, ma'am.
So we were talking about the cell structure of the prokaryotic organisms. So in that, what we have to discuss is the plasma membrane, which is the innermost layer, right? Now, this layer, whether it is prokaryotes or it is eukaryote, it is selectively permeable in nature. Selectively permeable means that it will allow only a few molecules to come inside and a few molecules to go outside, not every single molecule. And it is very similar in terms of its structure as compared to the eukaryote. So like the eukaryotic cell membrane and the prokaryotic cell membrane, they're very similar to each other. Now, in the case of the prokaryotic organisms, what happens is that there is an extension or sort of like an invagination of the plasma membrane. So if the plasma membrane is like this, then slowly it will start forming a depression and form a structure which is called as the mesosome. Okay, this is called as the mesosome. So it's like if this is the plasma membrane, what happens is that it will start depression, like a depression will be made like this, and then a deep pocket-like structure will be made, and that pocket-like structure is called as a mesosome. Now, these mesosomes, they can be found in terms of vesicle, tubules, and lamellae. They can exist in variety of shapes and forms. Talking about what does the mesosomes do, like what is their function? So, the mesosomes, they are responsible for the cell wall formation. So, they form the cell wall. Then, they are responsible for DNA replication and distribution to the daughter cells. So, it means that they help in sort of like a division process. Then they also help in the respiration, secretion, and since it's like a curved or you can say twisty structure, it increases the surface area of the plasma membrane of prokaryotes. Now remember that these mesosomes, they are not found in the case of eukaryotes. Is it clear? Okay, Sanidhi, have I done the introductory part of Animal Kingdom with you? The basis of classification and everything?
Yes, ma'am.
Okay. Now talking about gram staining. Now see, what is gram staining? See, depending upon whether the bacteria can take up a particular type of stain called as the gram stain, the bacteria could be of two types, gram positive, gram negative. Now, those bacteria that takes up the gram stain, we call them as the gram positive bacteria. And those who do not take up the stain are called as the gram negative bacteria. Now, when we talk about these bacteria, remember that the mesosomes, they are found in the gram positive bacteria only and no other bacteria. Only and only in the gram positive bacteria. Now, what is the difference between the bacteria that do take up the stain and the bacteria that do not take up the stain? See, the bacteria that take up the stain, they have a thicker cell wall or peptidoglycan layer in them. A thick peptidoglycan layer is present. Whereas the ones that do not take up the stain, they have a very thin peptidoglycan layer. And this gram staining is a staining technique to like divide the bacteria depending upon their ability to take up the stain. Clear? And it was developed by Christian Gram so as to classify the bacteria into groups. Any doubts?
No, ma'am.
Okay. Now, the bacteria, depending upon whether they can move or they cannot, the bacteria, they are of two types, motile and non-motile. Now, motile bacteria are the ones that have a flagella present. Flagella is the organ, or you can say it is the structure for locomotion. So, those bacteria, those cells that have a flagella present, they will be called as motile. Okay, they'll have this property of motility in them. Whereas the ones that do not have it present, we will call them as the non-motile. So the motile structure, they are the extension of the cell wall and they help the bacteria to move with the help of flagella. So talking about the flagella, so they are long filament-like structures and made up of three parts: filament, hook and basal body. Three parts make up a flagella. Now the hook portion is the portion that is embedded, or like the basal body is from where the flagella arises. Okay, so it's like most at the base of the flagella, and then begins the hook portion and the part of the flagella that comes out of the cell, that the part that is visible, the part that helps the bacteria to move along, that is your filament. So talking about the filament, it is the longest part and it comes outside the cell. Talking about the, like, see, remember that bacteria, they have certain surface structures, meaning those structures that arise from their surface. Okay, and that helps them to, you can say, perform variety of the process. So apart from flagella, there are other surface structures as well, such as villi and fimbriae. Now, when we talk about villi, these are like, they are made up of special protein called as pillin protein. Okay, so they are tubular structure made up of pillin. And what is their function? Their function is to help the bacteria to transfer DNA. So for example, if this is one bacteria, this is another bacteria. So in between them, sort of like a tubular structure will be made to transfer the genetic material from here to here. Understood? So this is the pili. The next surface structure is the fimbriae. Fimbriae is just like how you have bristles of the toothbrush. So just like that, bristles are present all around the surface of bacteria and we call them as fimbriae. What they do is, like, whenever the bacteria has to attach itself to some substratum like this. So with the help of that fimbriae, the bacteria is going to attach. Understood? Surface structures are clear? Any doubts?
Yes,
No,
no,
ma'am.
ma'am.
Okay, very good. So now moving forward, talking about the prokaryotic ribosome. Now see, ribosome is one of the most common cell organelle found in prokaryotes as well as the eukaryotes. But the major difference is the 70S, right? The prokaryotic ribosomes, they are 70S type. Now remember, whether prokaryotes or whether eukaryotes, the ribosomes are responsible for protein synthesis. Right? They are used for the process of translation, or you can say the process to make the proteins. Now, the size of the prokaryotic ribosome, it is 15 nanometer by 20 nanometer. That's the size. The prokaryotic ribosome, they are associated with the plasma membrane. Okay, there is no other part in the prokaryote cell that ribosomes will be present. So it is associated with the plasma membrane. And amongst that 70S, see, a structure, if you make a structure of a ribosome, so there is a bigger unit, which is called as the large subunit, and then there is the smaller subunit. Ribosome looks like this. So the larger subunit is 50S type, and the smaller subunit is 30S type. And altogether, it is 70S. Now, don't add them because that's never going to come to the conclusion. But remember that 70S is the general type of the prokaryotic ribosome. Individually, the 50S is the larger subunit and the 30S is the smaller subunit. Now, you people, do you know what is an mRNA? Achha, do you know what is an RNA? RNA, so you know, right? Like how DNA is there, same way RNA is there. Now, RNA, they are of variety of different types, like tRNA, mRNA, rRNA, sRNA, different types of RNA are there. Like tRNA means transfer RNA. mRNA means messenger RNA. So that messenger RNA, so if, for example, this is the messenger RNA and on top of this RNA, several ribosomes have attached themselves, like with the bigger and the smaller subunit. Several ribosomes attach themselves on the mRNA and the structure that is called as a polymerase. or polyribosome. Clear? So, when many ribosomes attach themselves on the mRNA, this structure is called as polyribosome or polysome. And what is the function of this polysome? It's that they will translate mRNA into proteins. Translate means, or the meaning of translation is protein synthesis. So, for the formation of protein, this ribosome attached with the mRNA is important. But this is not found in eukaryotes. Eukaryotes have a different mechanism. Clear?
Yes, ma'am.
Now talking about the inclusion bodies. Now inclusion bodies are the non-membrane bound structures, meaning they do not have any membrane, and they are usually spherical structures found inside the cytoplasm. And they, like, depending upon which type of inclusion body is there, they have different functions. For example, they are responsible for particularly storing the food material. So they store the reserve food material in the form of inclusion bodies. So there are different types of inclusion bodies, like there is phosphate granules, cyanophyshin granules, glycogen granules, different types of granules are there. Clear? Now, you know that there is blue-green algae, also called as cyanobacteria, when we studied Kingdom Monera, right? And then there is also purple and green sulfur bacteria. So these two bacteria, they are mostly aquatic in nature. So that is why they have gas vacuoles, which provides them the buoyancy, right? Which is the upward force. So it doesn't let them sink to the bottom of the water body. Understood? So with this, all the structures related to the prokaryotic cell are done. Do you people have any doubts up until now?
No, ma'am.
Okay. But don't worry, this was a very minor chunk of this chapter. The major chunk is now when we talk about the eukaryotic cell. It's in depth, and you know that eukaryotes are complex, so they have more structures, right? They have more cell organelles in them. So up until now, do you people have any doubts?
No, ma'am. No, ma'am.
All right. Apart from this, is this chapter easy?
So far, yeah.
Yeah, it is easy, isn't it? It's not difficult. Up until now, whatever chapters I've taught, which chapter did you people find the most tough? Like, you're like, ma'am, what are you saying? I don't understand. Like, how many
Animal
chapters
Kingdom.
have bounced off of your head?
Animal Kingdom.
Animal Kingdom. Okay, now you too have missed the classes also. That is why you feel like that. Other than that, like whatever you've attended, tell me about that.
Nothing as of now. So it's all zoology.
What about you, Sanidhi?
Ma'am, same. Nothing like how we have learned. I've not understood.
Good to know that. So it means the teaching is going right, right?
Yes,
You
ma'am.
people are able to understand the NCRT on your own.
Yes, ma'am. Yeah.
Very good. Are you people like reading the NCRT regularly?
Yes, ma'am.
Very good.
Yeah.
Keep that. Okay, even if you're not doing, it's all right, but you do that. Understood? Because it will help you. The more you read NCRT, the better you'll get. Trust me. NCRT for class 11th and for 12th. Make it your best friend. Remove your human best friend. Make NCRT biology your best friend. For 2 years, that's it. Uske baad you can make any best friend. I don't care. But for now, you make your NCRT your bestest friend so that it will never fail you. So tell me, what is the last thing you people wrote down in the previous class? I think it was mucus. This is the last thing.
Yes, ma'am.
This part only, glycocalyx.
Yes.
Yes or no? Okay.
Yeah.
Mom, you can scroll.
Yeah, both of you are done, students.
Yeah.
I'm done.
Both of you are done, students.
Yeah.
Mom, done. I'm done.
Both of you are done, students?
Yeah, ma'am.
I'm done. I'm done.
Both of you are done, students.
Yeah.
Any doubts?
No, ma'am.
No, ma'am.
Oh.
I'm done. I'm done.
Done, students?
Yes, ma'am, done. I'm done. I'm done.
Done, students?
Yes, ma'am.
Any doubts up until now?
No,
No,
ma'am.
ma'am.
Okay, very good.
Ma'am, done.
All right, any doubts up until now, students?
No, ma'am.
Okay. Now let us see the things about eukaryotic cells. Now you know that eukaryotes are the ones that have a membrane-bound nucleus present, and along with that, they have a membrane-bound, like all the cell organelles are also membrane-bound. So the members that come under the eukaryotic cells would be the kingdom protists, the kingdom fungi, plantae, and animalia. All these kingdoms right after Monera, all of them are eukaryotes. Now, eukaryotic organisms, they have a specialty of having superior cell organelles. They have superior characteristics, superior, you can say, things to exist as such. And apart from that, they have more complex structures in their cells. So each of those things we're going to see. Now, in them, a proper plasma membrane is there, and cell wall is present, but always remember that the cell wall is present in the plants, in the fungus, and some of the protists, and it is absent in the case of animal cells. Always remember this, no matter what happens. So now different organisms have a cell wall of different composition. So when we talk about the cell wall of the plants, it is made up of cellulose. The cell wall of fungi is made up of, you people tell me?
Ma'am, can you
These things
repeat?
the cell wall of fungi is made up of. I've taught you all of these things. No one.
I can't hear the question. Can you
The
please repeat?
question is, the cell wall of fungi is made up of what? Very bad, very bad. Chitin. The cell wall of fungi. Remember, I told you people to remember it as chitin? To remember the spelling? Or forgotten?
Yes, ma'am, I remember.
Ah. So
I
chitin, right? Chitin is what the cell wall of fungi is made up of. And then what is the bacterial cell wall made up of? Hmm?
cell wall of
Bacteria. No one is studying, huh? When do you people study? Yes.
ma'am, sugar and amino acid.
So what is that compound? What is the name of the compound? It is peptidoglycan. The cell wall of bacteria is made up of peptidoglycan. Now let us see the differences between the cells of the plant and the cells of the animal. So when we talk about the cells of plant and animal, comparatively, the plant cells are larger as compared to the animal cells. The plasma membrane in the case of plant cell, it is surrounded by a cell wall, which is made up of cellulose, whereas the plasma membrane of the animal cell is not surrounded by anything. So because they do not have a cell wall. Now, in the plants, they have a plastid present, which is chloroplast, like the different types of plastids are found, whereas in the animals, those plastids are absent. Most mature plant cells, they have a permanent and large central sap vacuole. So they have a big sized vacuole present in them, whereas the animal cell, they have, like they may have vacuoles, but always remember that their vacuoles are going to be temporary. It's not going to be a permanent and fixed vacuole, and most of the time their vacuoles are completely absent from them. Golgi apparatus of the plants, they are simple and you call them as dictyosomes, whereas the Golgi apparatus of the animals, they are highly complex, so it is like an apparatus. And lastly, the plant cells, they do not have centrosome or centrioles present in them, whereas animal cells have. So the mechanism of cell division in the case of plants and animals, it's also different. Understood, students?
Yes, ma'am.
Yeah, ma'am.
Okay, now note it down.
Ma'am, done.